With that, Croeso, good evening and welcome everyone. It's a great pleasure to be able to introduce an old friend this evening. So Daniel Davis is Professor of Immunology and Head of Life Sciences at Imperial College London. Interestingly, for a biologist, Dan obtained a doctorate in physics before going on to start his studies in immunology at Harvard University. His current research uses super resolution microscopy to study the immune system. He's the author of over 145 peer-reviewed papers that have collectively been cited over 14,000 times. He's a fellow of the Academy of Medical Sciences. Dan has also managed to be the author of 3 popular science books and is a regular at the Hay Literary Festival. His recent book, highly endorsed incidentally by the novelist Bill Bryson, is The Secret Body, and which will no doubt feature in this evening's lecture. His earlier book, The Beautiful Cure, was shortlisted for the 2018 Royal Society Science Book Prize and as Book of the Year in the Times, Telegraph, and the New Scientist. His first book was The Compatibility Gene, which discusses the genes and proteins that influence the success of transplantation, as well as the scientists who helped us understand the biology associated with these genes. Dan manages the difficult challenge of making his books accessible and enjoyable to both the layperson and the subject expert. His books are always meticulously researched and provide fascinating insights into the individuals who have made the scientific discoveries that have helped shape our understanding of the world. Dan, it's a great pleasure to have you to give this evening's lecture. Many thanks indeed. And we're very much looking forward to your presentation this evening.
Brilliant, James. Thanks so much for inviting me. It's really wonderful to speak to you tonight. Hopefully you can hear me all right. And moving forward in the slides, right, go. Okay, thanks so much. It's wonderful to be here. Imagine yourself as an alien with an exceptionally powerful telescope trying to understand what happens on Earth. You come across a football match, but your telescope isn't powerful enough to see the ball. You can make out there's a pitch, there's some players moving about, there's some sort of organization to what's going on, but it's really hard to understand precisely what's happening. And you just publish the observation in the alien journal of what's going on the planet Earth, and a few aliens email you congratulations. In time, alien telescopes improve. And then you occasionally see that sometimes one of the players falls over in front of the goal. And sometimes, but not always, that's followed by crowds of people around the pitch cheering and waving and shouting. It still doesn't make much sense. You still don't really understand what's going on, but this does lead to quite interesting discussions in the bar at the Alien Congress of what's going on the planet Earth, and you get your research funding renewed. Eventually, alien telescopes improve again. And then one of your colleagues notices something very intriguing, which is when the player in front of the goal falls over, then whether or not all the crowd start cheering seems to correlate with something else, which is whether or not the net bulges outwards. Now, this leads your colleague to have a really brilliant idea. She says that what if there's a ball there, but our telescopes just aren't good enough to see it? Now, at first, this sounds like a ridiculous idea because you can't see the ball. Your telescopes just aren't good enough to see the ball. But once you start having the idea that there could be a ball there, and then you start to think about what that would mean for how the rest of the things that you can see with the telescope work, suddenly it does start to fall into place. And now the game starts to make sense, that they're trying to get the ball into the net. It bulges. When it bulges outwards, then all the crowd start cheering. Eventually, everyone agrees with you that there has to be a ball there, even though you can't see it. And then, with everything now making sense, you start to win prizes, you, your colleague who had the idea, and the people that improved the technology to get you to the point to have the idea. Everyone wants to be your friend. And essentially, that is how a lot of science works, that there are advances in technology that show us more and more about what's going on. And from that, we have big new ideas about how things work, even if sometimes you can't directly see what it is that you're saying must be happening. It could be that the telescopes never managed to see the ball, but the ball must be there because there's a heavy weight of evidence saying that it must be there, and everything makes sense with it being there. I think that this is happening right now in our understanding of the human body. Technologies are moving forward quite dramatically, showing us new insights into how the whole human body works, and this is giving us quite radical ideas about how different parts of the body work on lots of different scales, right from how cells work to what mental health really means, to how different organs work, to the genetic codes that govern other aspects of who we are. All these different parts of the human body is are being opened up by new kinds of technologies. That's essentially what I wrote a book about, to describe how different technologies are giving us different kind of views of what we are and how this is radically changing how we see things. So I don't have time to go through every aspect of that, but I'm going to focus on, in the beginning, a particular story around new technologies that show us what cells are doing and how that leads us to different kinds of medical treatments. So Robert Hooke, when he was about 30 years old, published in 1665 the book Micrographia, which was using a very primitive form of microscope to show us things like this monstrous flea, previously just one black dot, and now this incredible monstrous organism revealed by the microscope. This was a hugely popular book. Samuel Pepys called it the greatest book he'd read. Everyone was very excited, and it's very clear that microscopes, as they advance, are one way of us seeing new worlds that we simply didn't know existed. So having a microscope that looks with better and better resolution is a surefire way to see new aspects of nature in general, and even specifically what the human body really is. But in 1872, a mathematician, Ernst Abbe, did a calculation about how good a microscope could be. From the way light is, the way that the fundamental nature of light, the way it bends around objects by a process called diffraction, that gives you a limit on how good any microscope, any light microscope can really be. And the formula that he used is now written in stone in Jena, which is a city in Germany where it's famous for developing microscopes and where Ernst Abbe worked. Roughly speaking, you can't just zoom in forever by building better and better lenses and optics. There's a fundamental limit, which is about half the wavelength of the light that's coming through a microscope. So sort of if room light roughly is a wave with a length about 500 nanometers, then a microscope can't see better than 250 nanometers. In other words, if two dots were sort of 250 nanometers apart, you couldn't easily resolve them as two dots. You might just see them as one dot. So there's a fundamental limit to how good a light microscope could ever be. So that puts a sort of stop, if you like, on the limit at which we could start to see new worlds happening in cells. But it turns out that microscope limit has been broken, and that allows us to see new things happening in the body that we didn't know were happening before. So to understand the journey by which we get to that revolutionary new kind of microscope and then how we use that for medical things, I'm going to tell you a little story as sort of how we get to that point. And really, the journey begins in mid 1970s. This is Osama Shimomura with his family, his wife and two children, who Akimi and his two children, Sachi and Sutomu, who were working on an island just off British Columbia looking for particular kinds of jellyfish. Now, Osama Shimomura was a Japanese-born scientist working in the USA, interested in how animals communicate by color. Lots of animals communicate with each other by the colors they have, and he was interested in the particular mechanism, the process by which animals could change and glow in their color. He focused on a particular kind of jellyfish that had green glowing cells at the rim of the umbrella part of the jellyfish. So around that rim there, this particular jellyfish had these green glowing cells. And he wanted to understand how was it that this jellyfish could glow green. So him and his family went down 6 a.m. in the morning to start collecting jellyfish, and they roped in their friends to collect all these jellyfish. These are photos I got from his daughter, who's now grown up, and Osama Shimomura is now passed away. But these are their friends. They're catching all these jellyfish. People would say to them, oh, you're catching all these jellyfish. How do you cook them? And they would say, we're not cooking them. We're going to use them for this scientific experiment where we're trying to understand how they glow green. Now, Osama Shimomura was a certain kind of scientist. His success comes from being very independently minded and thinking about how to do the experiment. So normally you might start a scientific process by reading lots of papers around a subject and thinking about what was done before and then proceeding with that kind of method. But Osama Shimomura was very independently minded. He said that a very important thing for being a scientist is to learn how to learn. He wouldn't just read the methods of other papers and follow them. So, for example, to separate out the green glowing part of these jellyfish, he would go down the local market, find some fabric that looked like he could filter things with. He made a sort of blender that was just essentially a kitchen juice blender to grind up jellyfish, put them through fabric, isolate the green glowing part. And it was very important to him that this was how you do science. You think deeply about what you've got to do and you find your own methods of how to do that. So these are the jellyfish he's studying, and he's trying to understand how you get green glowing cells at the bottom of that jellyfish. And he identified two proteins that are involved in that process. One emits blue light when it binds calcium, and then another molecule takes up that blue light and glows out the green light. And that second protein is called green fluorescent protein. So he published that, and obviously it has interest at some basic scientific level, but he's driven by curiosity about how jellyfish glow green, and he's not thinking about the practical application of his work, and he's certainly not thinking about medical applications of what he's doing. And there's a deep wonder here in how doing something seemingly just for knowledge's sake, how do the jellyfish cells go green, doesn't immediately, he's certainly not trying to cure cancer. And yet the thing that he discovered becomes used widely in medicine and really is quite revolutionary. And eventually Osama Shimomura is going to win a Nobel Prize for his work. So now we're going to jump to 1989. And there's a particular date in April 1989 where Martin Chalfie, a Chicago-born scientist, is in Columbia University in New York, and he is sitting in on a lecture where someone just happens to mention the green fluorescent protein that Osama Shimomura had discovered. And it becomes a light bulb visceral moment for Martin Chalfie, because he starts to think about how you could use that green fluorescent protein. Now, this was in the days, 1989, where you couldn't just Google what's going on about green fluorescent protein. So he starts to phone people, Has anyone heard of this green fluorescent protein? What is it? How can I get it? And he was led to phone another scientist, Doug Prasher, who was working in Woods Hole in the Oceanographic Institute. And Doug Prasher was isolating the gene that encodes the green fluorescent protein in jellyfish. So Martin phones Doug and says, when you get the gene for the green fluorescent protein, just send it to me because I'd really love to use it in some experiments. Martin then went on sabbatical. Doug tried to contact Martin, but he couldn't because he was on sabbatical, so he thought he just left science. Martin didn't hear from Doug, so just thought the whole thing fizzled away. So they kind of lost touch for a while. Eventually, Martin comes back and reads in a journal that Doug has now isolated the gene for the green fluorescent protein. So he writes back to Doug and says, "Oh, are you going to send it to me?" Doug says, "Oh, sure, yeah, I'll send it to you." Doug sends him the gene that encodes for the green fluorescent protein in jellyfish. Martin passes it on to Kirchin in his lab and says, why don't you just try seeing if this gene would work in another kind of cell? So Kirchin takes this gene from a jellyfish and makes it work inside a bacteria, puts it in a bacteria, and wonders whether the jellyfish gene would start to make the bacteria glow green. She looks down the microscope and no, the bacteria not going green. So the jellyfish gene doesn't make bacteria glow green. Oh well. But then she does think that the microscope in Martin's lab is quite old and a bit rubbish. So she double checks on another microscope down the corridor and it does glow green. This is what she sees under the microscope, green glowing bacteria. So this isn't entirely revolutionary in the sense that we know that genes, the basic way that genes are used by life is true for bacteria and true for jellyfish and true for human beings. So we know that genes could work in other cells, other kinds of organisms, but this is still groundbreaking because it wasn't really clear that one single gene, which makes one single type of protein molecule, would glow green. It could have been that other things were needed, the environment of the jellyfish, there's something else in the cell of the jellyfish. It could well have been that didn't work. It turns out that it did work. So Kirchin was the first person in the world to see that the green fluorescent protein from jellyfish could make completely different kinds of cells also glow green. So the next part of the story is done by another person in Martin's lab, Yuan Tu. And this time he puts a tag on the green fluorescent protein gene so that it should only come on in particular touch sense neurons of a certain type of worm, C. elegans worm. And this is what he sees. So that the worm now has specific green glowing cells inside it. So now a particular type of cell could glow green inside another organism. They published this first of all in 1993 in the Worm Breeders Gazette, which isn't really the kind of place that you normally publish a Nobel Prize winning experiment. But eventually they do publish it in Science, which is an extremely prestigious scientific journal. But initially it's in the Worm Breeders Gazette, and they just say, we've got this gene that makes particular neurons of a worm glow green. If anyone else wants to use this gene, just let us know and we'll send it to you. Martin Chalfie, when I asked about this, in hindsight, it seems quite revolutionary that you could make a particular cell inside a complicated organism glow green, because you could then watch what that individual cell does in the whole organism. Why is it then at the time people didn't really realize that was entirely revolutionary? And Martin says that the thing is, when you discover something really different, really out there, as it were, different, it takes a lot of time for people to get used to the idea. You have to tell people lots of times. When Martin told people in Columbia University, it was really exciting. I've got this worm with neurons that glow green. He says that people weren't very excited about it because you have to tell people lots of times so they get used to it. One person who he did tell lots of times about it was his wife, Tula Hazelrigg. And she was also an eminent scientist in Columbia University, and she really did the experiment that was truly groundbreaking in how we use this molecule today. The green fluorescent protein looks like this. It's this sort of barrel-shaped structure, and the middle part of it glows green. And her idea was to, her idea. Well, here's Tula Hazelrigg. This is later after Martin wins the Nobel Prize. But her idea was to attach the green glowing protein onto another protein molecule. So you could do that just by making the gene for the green fluorescent protein literally read into the gene of another protein molecule, and then you would produce a green glowing version of any particular protein molecule you like. So now she wasn't tagging a whole cell, she was tagging an individual type of protein molecule, a biological laser pointer, as Discover magazine used it. And this is exactly how we use GFP today very commonly. So we put the gene for GFP with the gene of another protein, and now we make a green glowing version of any protein we're interested to see what's happening inside a cell or in an organism. Roger Chen was a scientist who made other versions of the green glowing protein. Coral, for example, produces a red fluorescent protein. Then you can make different colors of different molecules. And Martin Chalfie and Roger Chen won the Nobel Prize for using GFP. But Doug Prasher was not awarded the Nobel Prize. And when Martin won the Nobel Prize, at that time, Doug Prasher, remember, he was the person who made the gene for green fluorescent protein and sent it on to Martin Chalfie. Doug Prasher lost his job at the Oceanographic Institute in Woods Hole because things changed there. He didn't get his funding. He then had a job at NASA, but NASA changed their priorities, so he lost his job there. He had a year of unemployment, and in his own words, he suffered bouts of depression. And at the time that Martin won the Nobel Prize, Doug Prasher was working for about $10 an hour in a Toyota car dealership in Huntsville, Alabama. He said that he didn't want to. He went back to live. He didn't want to move cities because his daughter was, in a high school and didn't want to disrupt his family life. So at the time that the Nobel Prize was celebrated, Doug Prasher was just working in a car dealership. Martin got in touch with Doug and paid for him to attend the Nobel Prize ceremony. And Doug says that he's not sorry about what happened because it was Martin and others who used the green fluorescent protein. So he's proud that his work was used. Okay, remember we're on a journey to create a new kind of revolutionary microscope that sees better than physics really allows. And so far we've talked about jellyfish and making worms glow green. So now we're going to come back really to how this led to the microscope. And now the story comes to Eric Betzig and Harold Hess. Eric Betzig and Harold Hess worked in Bell Labs, which is a very driven, cutthroat, high energy environment, famous for the discovery of the laser and other things. But when Eric Betzig was age 42, he was burnt out, had enough of science, thought that people were sort of overstating what they thought they discovered, and he'd had enough of it. So he quit, and he was at home with his family. He did start to think that he missed science, and he met up with Harold Hess in national parks, his old friend from the Bell Lab days, and they used to talk about how they did miss science, he did miss science, and how he'd like to contribute. One day when he was out walking his daughter in a stroller, he had a big idea about how to build a new kind of microscope which could beat this fundamental law of physics. He then told Harold Hess that he had this idea of how to build a new microscope. He published it in a journal, just the idea itself, and left it at that. But when he read about the green fluorescent protein story, he got back in touch with Harold and says, we've got to build the microscope. We've got to get back and do this, because now you can make a green glowing version of a specific protein molecule. And he's got the idea of how you could build a microscope to get down to that kind of resolution that becomes really exciting. So they get back together and they decide they're going to build the microscope that Harold has had the idea for, that beats a fundamental law of physics, essentially. They built it. They both spent $25,000 of their own money. And Harold Hess says, well, you could spend that money renovating your bathroom, but what's the point of that? They built it in Harold Hess's living room. And Eric Betzig said they did that because at the time Harold Hess wasn't married. And they worked day and night. They did use some parts from Bell Labs as well. And they rekindled the sort of spirit of the Bell Lab work ethic. Sometimes one of them was sleeping, the other one's carrying on working, and they built the microscope. This is the microscope that they built in Harold Hess's living room. It took them 6 months or so to build it, and they proved that it worked. Now, how it works exactly is this. This is the microscope that beats a fundamental law of physics. You can't naturally just zoom in with better optics. So if you take a picture of a cell, which is at the top here in the sort of splodge of image with lots of dots, then as you zoom in and in and in on these individual protein molecules, you get a blurred picture of what you see below. Eric Betzig's brilliant idea was that if you just use laser light to flash at a relatively low level and just get a few of the molecules to switch on and glow green, you don't turn them all on, you just switch a few of them to glow green, then you get a splash of light where one molecule is now fluorescing out the green color. Even though that flash of light, you can't see an individual molecule, you know that big splodge of light has come from one individual protein molecule because you've only turned a few of them on. And so you could put a dot right in the center of where that flash of light is happening. And then that one would go off, and now another one would come on, and you could put a dot in the center of that big flash of light. So you just excite a small number of molecules at any one time. And because by chance they're not going to be overlapping, you know the big flash of light you see is coming from one molecule, and it's coming from in the center of the big flash of light. And you do that. So you put a dot in the center. You do it again. You put a dot in the center. And then you build up a single molecule picture of where everything is at a resolution you could never have done before. And well, there are two things that I find really inspiring about this. Firstly, it's the guts to think that a fundamental law of physics doesn't really mean you couldn't build a microscope that could beat it. It's having that bravery and that courage to think maybe I can think of some way around this. And secondly, it's about thinking about tricks. It's not about thinking that the fundamental law of physics is wrong. There's nothing here that is changing physics. It's a trick. It's a bit like how an aeroplane doesn't change the laws of gravity. If you just think of a trick by which you could get such a large weight to fly in the air, which is about the wing shapes, of course. And it's the same kind of thing. He doesn't think gravity, the laws of physics must be wrong. He's thinking of a trick by which you could build a microscope to do better. He realizes that he has to really prove this not in Harold Hess's living room, but in a proper scientific institute. So he works with Jennifer Lippincott-Schwartz in NIH to prove that this microscope works. And she deserves great credit as well, because if someone tells you, I'm unemployed and I'm at home and I've built in my friend's living room a microscope that beats a fundamental law of physics, it's not clear that you would say, okay, let me try that out in my lab. So Jennifer Lippincott-Schwartz also deserves credit for understanding there's potential here, encouraging Eric Betzig and Harold Hess to build the microscope in her lab in the NIH to really test it properly with cells. And this is the version of the microscope that they then built in Jennifer Schwartz's lab to test things out properly. It took them a few months to go from here to everything that they needed to essentially for Eric Betsy to win a Nobel Prize for discovering and the microscope that changes this fundamental law of physics. So I'm sorry, that beats this fundamental law of physics. The way I'm telling the story is that it's highlighting the sort of a few of the individuals, but actually it's also true that there were other people at the time doing similar related things, and it's not. And I mean, obviously that is important, and in my book I describe that more. But essentially, it's not quite as linear as the scientific hero stories often make out. There's much more of a network of things happening and ideas germinating in different places around the same time. This is the version of this kind of microscope that we have in my lab now. We have 3 of them in my lab at Imperial College London, and they cost about £1 million each. And now we're using them to try to understand how cells work. We're looking at the immune system in particular. This is a cell straight out of your blood, which is the cell on the right, and it's an immune cell, and it's got to decide whether the cell on your left is healthy or diseased. And we use these kind of microscopes to watch this process happen. One of the first things that I discovered now many years ago is that a lot of the protein molecules that are important for the immune cell to decide if this other cell is healthy or diseased move up and accumulate at the contact between the 2 cells. So that green splodge of light you see in the middle is a green fluorescent protein tagged molecule which moves up to the contact between the 2 cells, and that's important to how the immune cell knows whether the other cell is healthy or diseased. It turns out then that if you look at that interaction in 3D, you have different protein molecules that move to different places across the contact between the 2 cells. And I saw that when about 20 years ago now, when I was working in Harvard University, and that was reminiscent. It reminded people of what was known to happen between neurons. So it became called the immune or the immunological synapse. Now, if the immune cell on the right sees this other cell as a disease cell, in this case it is actually a cancer cell, then it takes those cues from the molecules that have moved up to the middle, and now it's going to kill the cancer cell by by those red blobs are blobs inside the immune cell that contain toxic molecules, and they're going to move up to the contact between the 2 cells, and then the contents of the red blob is going to come out, and it's going to cause the cancer cell to die. This is a picture of the cancer of the immune cell, as if you were looking at it as if you were the cancer cell. So if you were the cancerous cell and behind the screen is the immune cell, this is the sort of interface, the contact point. And this is just below the surface of the immune cell. There's this dense structure, which is called the cytoskeleton of the cell. So this is underneath the surface of the cell. There's this web of proteins that normally are there. They allow cells to move, and they allow cells to. They're important in how cells divide. All sorts of processes that are dynamic for cells is controlled to a large extent by the cytoskeleton of the cell. This meshwork of proteins. Now, so if you're the diseased cell, you're looking at this, and behind the screen then would be the immune cell. Now, those red, if the immune cell's going to try and kill you, and the red blobs that are behind the screen have got to come through this meshwork to then release their toxins to kill the diseased cell. And what we noticed was that for this to happen, the holes in that meshwork of stuff, if you imagine that as like a meshwork, like the inside lines of a tennis racket, then to allow those red blobs to pass through and deliver the kill, they have to move apart and get bigger. So if we colour them according to their size, the holes get bigger. Now, we could have only seen this with the revolutionary new kinds of microscopes. You could not have seen this process happen with any normal light microscopy until these groundbreaking new microscopes were discovered or invented. That's how we could see this process. So we see this new thing that cells are doing only because of these new microscopes. So essentially, there's this meshwork of proteins, and the holes have to get bigger to allow these toxic red blobs to come through and then kill the diseased cell. So that's a fundamental process of how immune cells can kill off virus infected or cancerous cells in your body. And we only really got to see that process, how it works, with these new kind of microscopes. Now, my dad died last year now from multiple myeloma, but for about 15 years he was kept alive through taking a drug called lenalidomide. And lenalidomide is a medicine given to many patients with multiple myeloma, and it's based on the other drug thalidomide. Now, thalidomide you'll know about as being responsible for one of the worst tragedies in all medical history, when it was given to pregnant women, and untold numbers of tragedies happened, including many babies dying. It was anecdotally noticed, though, that thalidomide, even as well as having these tragic consequences, also did seem to have some anti-cancer properties, some protective property against people getting cancer. And so a drug company made a less toxic version of thalidomide, slightly different structure, and that has become the drug lenalidomide. But because it was discovered in that way, that it was just noticed that it had an anti-cancer property, it wasn't designed to have a particular way of protecting against cancer. It was just noticed that it did that. Then nobody really knew how it worked or what it does. So because my dad was taking this medicine, I thought, well, let's look at that in the lab and see if we can look at what the drug is doing for how the immune cells might be able to respond to cancer cells. And one of the things we noticed then was that when you treat cells with that drug that was used for multiple myeloma treatments, then what happened is that meshwork of proteins that have to open up to get bigger holes to allow the red blobs to come through, that happens much more efficiently in the presence of this drug, which fits with the idea that the immune cells were better at killing disease cells when they're on this drug. And in these different columns, what you're looking at is the meshwork of proteins. And when the immune cell isn't triggered to kill anything, nothing happens. When it is triggered, when it's stimulated, the holes open up, you'll get the red blobs coming through to kill. And when the presence of the drug, that plus sign on the last column there, the holes open up even more to more effectively allow the killing to happen. So that new kinds of microscopes showed us a process that happens, and then that led to an understanding of a mechanism about one of the ways in which this particular drug works. At a conference, I happened to be chatting in the bar to another scientist, Konrad Kuchevsky from the NIH in the USA, and he was working on something completely different from me, a specific genetic deficiency that led to children having lots of recurrent infections and problems, and they sadly often die quite young. And he then developed cells in a lab dish that had that same genetic alteration, and was studying what happens when cells have that genetic alteration that gives this problem to children. And he showed that one of the consequences of this genetic deficiency is that the immune cells in children are not very good at killing diseased cells. And this is just an example showing you from patients that the ability of them to kill a particular cancer cell is not very good compared to how good the killing is from normal blood cells taken from a normal healthy individual. He noticed that in the cells that had this genetic deficiency inside the immune cells, those red blobs that contain the toxic molecules to kill are much bigger than normal. He did an analysis to show that the types of things in those red blobs are actually quite similar. They still contain the toxic molecules, but the genetic deficiency had changed something about how that toxic packet of proteins develop, and you get much bigger vesicles, much bigger compartments of them being stored inside the cell. So this led us to the idea of maybe one of the problems is that the immune cell can't kill a virus infected cell very easily because the red blobs are big and now they can't get through that meshwork of actin. They can't get through the holes. I said, well, we know that this cancer drug actually opens up that meshwork more. So maybe that would allow these red blobs to come out much more easily. We couldn't test that in children because I'm not a physician, I do very basic science, but we could take cells from children and could show that, at least in a lab dish, that cancer medicine could slightly rescue the ability of their immune cells to kill diseased virus-infected cells. So what we got was new kinds of microscopes, showed us a new process inside cells, which led to the idea of how a drug works, which then led to being able to use that drug in a completely different setting. It's not that we can use that immediately to help these children because there could be other effects happening in people, but it at least shows you that in a lab dish, it's one of the things that could help. Now, I realise that we have not that long left, and I have several more things I'm excited to tell you about. So I'll have to speed up very slightly here, and bear with me because I realise that we are due to have some questions and things soon. So one of the other discoveries that was made with these kind of microscopes and other techniques is that when an immune cell sticks to a cancer cell and it's going to kill it, or it can be a virus infected cell, then the receptor molecules that are on the surface of the immune cell often get chopped off from the surface of the immune cell after some time. And people thought, well, that is because you need your immune system to calm down. Once you've killed off a virus or whatever, or you need your immune system to come back to its normal resting level. And so the immune system has lots of ways in which it relaxes back to its normal state, not in this heightened state. And one of the things might be to chop off the molecules that see cancer or virus infected cells. But that's not so useful. If you want to help your immune system fight cancer, you don't want those molecules to be lost. So drug companies were designing things to keep these molecules from being chopped off. But by using our microscopes, we discovered that the complete opposite thing was happening. That in fact, losing the receptor molecules that see a disease, losing them, helps the immune system. It's completely counterintuitive. And that's because when an immune cell sticks to a cell, let's say it's a virus-infected cell or a cancer cell, and then it gives across those red blobs, and then it kills the cell, it's got to remove all the molecules at that contact that let it see it was a disease cell, so it could detach and then go and attack another diseased cell. So losing the molecules that see the disease are important, helping the immune system fight disease because it goes from one cell to the other. And hopefully you can see that here. This is a natural killer cell, one of the cells out of your blood that is then going to move around and kill this cell, then it's going to kill this cell. It goes red when it's died. Now it's moving across and killing this one, red, then it's died, and then it goes down to that one, red, then it's died. That is one of your immune cells moving from one cancer cell to another one, to another one, to another one, killing them all sequentially. And it's doing that by losing the molecules. Each time it sees one, it loses those molecules, it can then detach and go and kill another one and another one. New directions in science are very often launched by new tools, much more than new concepts. It's the new microscopes that led us to do these discoveries. Another new kind of tool that we have is to study cells in a very, very deep way. and this leads us to discover entirely new kinds of cells. If we just have two labels on a cell, we can look at the groups according to what is this cell? Does it have some of A and B? Does it have some of B but not A? Does it have A and not B, etc.? And we can plot that on a diagram like this, and now we can identify types of cells out of this complex population just by looking at two markers. Now we have the ability to analyze what's happening to all the 20,000 human genes inside a cell, which ones are being used, which ones are not being used. And although we can't easily visualize that, a computer can calculate for 20,000 markers, just like we have here, and come up with clusters of cells. This group of cells are quite similar because they're using the same kind of genes. This other group is using the same kind of genes. And if we look at one type of immune cell here, this is work from my own lab then, we now get an analysis. What was previously one name of an immune cell is now lots of different types of an immune cell because we're analyzing what genes are being used and doing a complicated calculation. So it used to be with a primitive microscope, you could look down it and you would discover bacteria or sperm or a new kind of cell. Right now, today, we are discovering new kinds of cells, but in a different way. This is the equivalent of an analysis that leads us to see an entirely unexpected new kind of cell. Nasal swabs we're all very familiar with now because of the pandemic. And at the end of the nasal swab, what are the cells that are there? You do this kind of analysis, and we can discover the types of cells that are inside the nose, and we can start to analyze which ones of them are infected with COVID. How might that relate to the eventual outcome of a patient when they experience a particular type of symptoms from COVID? This is Ms. Hanifa, who is a dermatologist working in Newcastle. Instead of thinking about skin as you and I know it, she's looking at skin like this, a clustering of dots on a map. These are all the different kinds of cells that make up human skin. And this level of detail is unprecedented. And it'll be in her eyes, as a medical physician, as a doctor that you would go and see, she says that it's not too far in the future that you might go to your doctor and instead of looking at your skin, what's red, what's bumpy, as well as doing that, we might be able to do a very deep analysis from a small sample and analyze very precisely what all the cells are doing in your skin. In my own lab right now, this is the new kind of experiment we're doing. We're analyzing what cells make up you, make up the human body, make up your immune system, and we're doing it in yet another new kind of way with these super resolution microscopes. We have a cell landing on a glass slide that's coated with a molecule that makes that slide look like it's a cancer. So it's stimulating the immune cell to think it's cancer. We leave it for a bit. The cell then sends out those red blobs as if it's trying to kill a cell, but it's really just landing on our slide. We then add another color to mark everything else on the slide, and we take away the cell, but there's now a gap, a shadow, if you like, of where that cell landed, and we look at all the stuff that cell sent out in response to seeing the stimulation of a signature of a cancer. It looks like this. So we have cells landing on a slide, we take the cells off, and we look at what are the stuff that it's sending out. So in the top row, there's nothing there because we didn't really stimulate the cells. And in the second row, those are the red things, the red toxic molecules that are coming out of the immune cell to pretend as if it's going to kill the cancer cell. But the thing is, it's not just the proteins that we know about. Other things are coming out of these cells that we don't really know about. And so now we're analyzing what is each individual immune cell sending out in response to seeing this or that signature of a disease. And that leads us to this kind of really complicated analysis where we're looking at the individual cells. What do they do? We're characterizing the cells according to the types of things that they send out in response to a cancer cell. And that leads us to another way of defining new kinds of cells in the human immune system according to what it is they do, what it is that they send out, in response to seeing some signature of cancer. I think that there's an enormous revolution happening, hidden away really inside labs where technologies are advancing. They're giving us new insights into how the body works. I only really spoke about cells, but in the book I also discuss how it works for things happening about embryos, organs, tissues, the brain, the microbiome, and the codes around genes and other things that can parameterize who and what we are. This is not a small thing that's happening. This is a really big sweeping change in how we view the human body. I think in 1887, Heinrich Hertz built on Maxwell's equations, and he discovered the waves that we couldn't see. That light is a wave, but he discovered there are also radio waves and X-rays, other kinds of waves that we couldn't see. He had no idea that that could have led to the internet or the TV. I think that that's where we are right now in biology. Technology is advancing. We're seeing new things that cells do, new ways in which cells, even new kinds of cells, new ways in which the brain works and in which aspects of health can be defined. And it's as groundbreaking as this. And the implications are as hard to predict as this. You can't tell where this is going to lead, but I think it's as big as waves we can't see that lead to the TV and the internet, and we can't see where this is going to lead to right now. But for sure, it's going to change medical practice, because already now you can do deep analytics of your cells, of you, in lots of different ways. One day, this idea, a watch that tells you a few parameters about you, your weight, your height, your blood pressure, that's going to seem incredibly primitive. There'll be any number of analytical tools you could use to understand yourself. And the thing that's different about this revolution to many others is that you personally will be equipped to understand you, and it's up to you how much you use it. Things like the Industrial Revolution affected us in a sweeping way because we're all affected. There's nothing we can do individually. It's about the whole of society changing. This revolution about biology is happening, and it's affecting you personally, and it's up to you as to how much you will use things. I think we're still in early stage of where this is, of what things are happening, and what all of this, the secret body, means for our lives is still very much in the balance. I want to end with just one short thing. I know that we're short of time, but I want to end with one very short thing, which I just want to read to you very briefly. It's something that inspires me deeply, and so maybe it will mean something to some of you. When Eric Betzig, who won the Nobel Prize for the revolutionary kind of microscope, when he did his speech for the Nobel Prize, most Nobel Prize winners end by thanking all the people that got them to where they are, and Eric Betzig did that. But then he said something else, and I want to end by saying that to you. The last thing I would like to say is about taking risks. People are always exhorted to take risks, and that's fine, but you're hearing that from the guys whose risks paid off. It's not a risk unless you fail most of the time. And so what I'd really like to do is I'd like to dedicate my talk to all of the unknown people out there in any walk of life who have gambled their fortunes, their careers, and their reputations to try to take a risk, but in the end failed. I'd just like to say that they should remember that it's the struggle itself that is its own reward, and the satisfaction that you knew you gave everything you had to make the world a better place. Thanks for listening.
Thanks very much indeed, Dan. That was absolutely fascinating tour that you provided us with there. I'd like to request that our audience this evening maybe put their questions in on our Q&A. So please feel free to post your question on the Q&A. Dan, if I could just start things off, it's intriguing that many of the advances have been made from individuals, from inventors of new instruments, as you said, and it's intriguing that many of these seem to have been done on almost like a low budget to begin with, Shimomura and the guy with the super resolution. I mean, so you think there's still a place, even with the way science is structured today, for such individuals to make inventions that are going to transform our lives?
I think so. I think, there's lots of different ways in which science works, and sometimes it's true that there's a big budget is needed, and it needs to be something that everyone agrees on, and or at least has a chance of working, and it works through the structure of science with our grants and our budgets, some of it funded by government, some of it funded philanthropically, some of it funded by charities with a particular mission to cure cancer or diabetes or some particular illness. But it's equally true that sometimes it if you said to someone, I was going to make a machine that could see through the human body, no one's going to fund that. But there is an X-ray machine, right? So it is true that we need to allow things to happen in a slightly wild way to allow innovation to really grow. And that is a really important culture, that we need both things. We need people that are working very directly on curing diseases, and we need people that are working and we need people working on very curiosity driven things. Someone's just asking me to stop sharing. I think I have stopped sharing, have I?
Yeah, yeah.
Okay.
Sorry, Dan. Yeah, please carry on.
No, that's fine. That's fine. So I think we need all of it. We need people with an organized program of science, like the Gantt chart, like we're going to land someone on the moon. That takes a lot of organization, a big program, and we need to fund curiosity-driven research, which is very explorative, inherently explorative, and may stumble across something groundbreaking.
Great, thank you. Dan, picking up one of the first questions then, how long do you think it'll be until a cancer cure will be found? So maybe you could comment more widely on the excitement that currently exists around immunotherapy, and how
Sure.
long it might be from some of the observations that you and colleagues have been making before that's going to be translated more widely in medicine.
Sure. Well, almost certainly there will not be a cure of cancer, because cancer is many types of disease in its detail. Like most, it's like mental health or physical. Everything is very complicated, and so there's always going to be lots of cures applied to these illnesses, depending on the particular nature of the version of cancer a person has. There are great steps forward, though, in, as James mentions, using your immune system to fight off cancer. That includes the idea that so the immune system, it was discovered that the immune system has brakes on it, in that after a virus has been eradicated from the body, then the immune system has receptor molecules that act as brakes to switch off the immune system, because once you've eradicated a flu from your body, you don't want your immune system in this heightened state. Your fever has to come down. You have to come down and relax. But if your immune system is doing that while trying to fight cancer, that's not good. So medicines that prevent those breaks from coming on have proved to be a very important type of medicine for fighting cancer. But of course, it works for some patients in some types of cancer. And so part of the advances are in trying to understand who responds well to which type of therapy, so that we can target those more effectively. And there are other types of immune therapies on the horizon too. So I think there are six cases of a license being granted for the type of therapy where you take the cells out, immune cells out of a person, you give them a new gene that makes a protein that sees their particular cancer, and then you put it back in the patient, a so-called CAR T cell, and that now can work again for some patients in some situations. Now, there are many issues here which are important to bear in mind. I'm going to just say one, which is that as we advance these new kinds of medicines, I think something we need to be thinking about in society is how widely is this going to be applied? Those types of therapies are inherently expensive, and it is something of an existential crisis for the NHS, the model of the NHS, which we're all so proud of. What do we do when the therapies are really, 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 really expensive? And it's not the case that the pharmaceutical companies are just charging tons of money. These are complicated processes that have to happen for a patient individually. It is inherently expensive, and we need to be more open about what are we going to do to keep this up.
Yeah, thank you. Maybe following up on that then, Dan, as we personalize medicine, clinical trials become more challenging as we're dealing with smaller and smaller numbers of patients in each group. Do you have any thoughts on how we can deal with this?
I do agree that personalized medicine is a challenge, and it's true that I mean, obviously, people set up those trials so that there would be enough people in the studies that becomes a statistically valid test. But it's true that there are lots of complications there, and I think a lot of the time right now, we tend to think of science as being very precise and giving us a very precise answer. Um, uh, there are any number of examples of this, like how much should you sleep, or will anything where there isn't a precise answer because it's very personalized, and it's hard. It's hard. The science is hard. The science itself is quite hard to do. So although each of us as individuals want these answers very declaratively, and we encourage the media to keep giving us sound bites like, if you do this or that, it's really good or bad for you. It's true that the science itself is quite hard and it varies for each individual. It is a really big frontier as to how we understand human diversity. And yeah, I mean, I did write about this in my first book, but it's really important. I think it's the biggest thing for us to try and tackle, that we have to embrace the diversity of people for what we are and trying to understand all these issues. And so I suppose be mindful of sound bites, because very often it's impersonal and everything is personal.
Thank you, Dan. So Dan, in your book, The Secret Body, you talk about a number of different sort of areas of medicine, the importance of the microbiome, the microbiota, the organisms living in our gut, especially. You talk about the developments in the brain. So that leads on to one other question that we've got here. What specific medical breakthrough do you think would be the most impactful this century?
I mean, it's very, like I tried to say at the end, that it's very hard. There's no way that you could have predicted the discovery of waves we can't see would lead to the internet. I mean, it's mind-boggling to think that, right? And I genuinely think that something like that is impossible to predict will happen in the 100 years of this century, right? That thing is impossible. All you can say is things that are sort of on the horizon now, but you can't get as an imagination to the extent that I could say, well, the internet's coming, the equivalent of the internet for medicine. So I think it's very, very hard to predict. But there are many areas of health and science where our understanding is exceptionally weak, and more will be done. We saw that during the pandemic. We don't really understand what led to this diversity of symptoms, from hardly anything happening to a person to them having very severe consequences, and many people dying, and some people suffering from things like long COVID. We don't really understand what causes or what underlies all that spectrum. So there are many, many, many other areas. For example, how nutrition relates to immune health is very, very poorly understood. Although I see your next speaker, Tim Spector, may have some insight in that.
Yes. So, Dan, I think that probably brings things to a conclusion this evening. I'm conscious of the time. I know we've had a number of anecdotal comments coming in indicating how much our attendees have enjoyed this evening's presentation. Someone was particularly pleased to hear that failure is okay and is it'll be meaningful. So
Definitely.
I'm sure your talk this evening will be an inspiration for a number of the younger members of our audience this evening as well. So it just remains for me to thank you once again, Dan, for giving up your time this evening. I look forward to seeing you at the Hay Festival again at some stage in the future. I hope you got another book lined up at some stage. And I'd just like to again highlight to our audience that we will be sending a short link to you very soon via Mentimeter, whereby you can hopefully provide us with feedback for this evening's lecture. And just to remind everyone again, then, our next lecture in the series, as Dan has highlighted, will be given by Professor Tim Spector from King's College London, where Professor Spector is going to be talking about the future of personalized nutrition. So that's Thursday, the 29th of February, if you'd like to register for that lecture as well. So stay warm, everyone. Much appreciated again, Dan. Thanks again indeed. Good night,
Brilliant.
everyone. No start. Good night.
Thank you, James, and everyone listening. Thanks so much.
Bye.
Bye.